homologous recombination occurred in the wrong sites and caused cancer. And there was a successful gene therapy for the excellent adenoleukodystrophy. And they used the lentivirus vector, which is a HIV derivative. So in that X-linked adenoleukodystrophy, what happens is it's missing an enzyme that metabolizes fatty acids, and that fatty acids make aggregates, and then they mess up the neurons in the brain and in the peripheral nervous system. So it's the same thing over here. In this disease, you have the ADA, which is a adenosine deaminase enzyme, and here it's a different enzyme. Uh, in uh, in this one, in this disease, a different enzyme that's going to be added. So RNA interference. Okay, so this one is going to be. This is about. This is about this one over here. We said we'll talk about it later. So it's about this: how to mess up a gene that's toxic or causing a disease. That gene you wanna stop. So the RNA interference. They're double-stranded RNA. That's a key point. Double-stranded RNA, and they're 20 to 30 nucleotides in length, and they're derived from a longer base pairs, and they are cut by dicer in the cytoplasm, so they can silence endogenous genes or parasitic genes, or the pathogenic invaders such as HIV virus can be silenced. And so there's two ways: you can destroy the mRNA or you can inhibit translation. And in humans, we only found inhibition of translation, but we can cause destruction by artificially inserted interference RNAs. So after it's cut by the dicer, one of the two single strands is chosen by the RISC, and then it also combines with organoid, uh, which is a catalytic component, and it's a endoRNase, so it's going to cut up the messenger RNA that is going to be silenced. Yeah, so this one over here is so the previous thing I was talking about was from here, and this is the picture of what I was talking about. So you have the microRNA in this nucleus that has a strong hairpin formation. It has a cap and a poly A tail, and then it's going to come out. It's going to get diced by the dicer, and then you're going to have a single strand, and that's going to complex with the RISC. RISC stands for RNA induced silencing complex, and then it's gonna also gonna combine with organoid, and then you're gonna have the mRNA cut up, the one that's so rapid mRNA degradation. So now you have two main categories of RNAs, Si and Mi. Si is short interfering RNA, and mRNA is microRNA. And sRNA is exogenous precursor, and it can originate from natural introduction in the cell, like viral replication, and also artificial introduction through mediated in gene therapy. And microRNA are encoded on the genome. So here, what we can do is we can take the siRNA, double-strand siRNA, shake it up with some fatty acids, so it's coated with fatty acids, and then you can insert it into a mouse, and also, so this siRNA is gonna be complementary to the mRNA in the herpes virus. So over here we see two graphs, four graphs total. So this first graph, the red one, it's the herpes virus is on by itself, and 80% of the mice died. And then over here we have the GFP siRNA plus the herpes virus. So the GFP siRNA is just a control method to see if the GFP siRNA is actually causing any deaths. And it's not really doing anything, so there's like a 5% difference between 80% and 75%. So 75% died, so 5% less died. That could be just uh, that could just be like experimental error. That could be explained with that. And then over here, so we see the HSV siRNA. So this is going to be silencing the herpes simplex virus. The siRNA is going to be silencing the HSV, and they have two different siRNAs, 1 and 2. So we can see the number 2 is working better, because over here 40% died over here and only 20% died over here. That's about it.